Warmest greetings to all my incredible subscribers and new viewers alike. Welcome, welcome, welcome. We're thrilled to have you here as we tackle Dytiscidae. The Dytiscidae, from the ancient Greek word dystikos, meaning able to dive, are the predaceous diving beetles, a family of water beetles. They occur in virtually any freshwater habitat around the world, but a few species live in terrestrial habitats such as among leaf litter. The diving in their common name comes from their cycling between underwater and the surface to replenish oxygen like a diver. The adults of most are between 1 and long, though much variation is seen between species. The European Dytiscus latissimus and Brazilian Bifurcitus ducalis are the largest, reaching up to 4.5 and respectively, although the latter is listed as extinct by the IUCN. In contrast, the smallest is likely the Australian Limbodessus, atypicali of subterranean waters, which only is about 0.9 mm longitude. Most are dark brown, blackish, or dark olive in color with golden highlights in some subfamilies. The larvae are commonly known as water tigers due to their voracious appetite. They have short but sharp mandibles, and immediately upon biting, they deliver digestive enzymes into prey to suck their liquefied remains. The family includes more than 4,000 described species in numerous genera. The oldest of the species is Palaeodytes gutta, from the Late Jurassic according to Karabastau Formation fossils. Species employ diverse techniques and traits to source their oxygen underwater. Dytiscidae are adept swimmers, thanks to their enlarged, flattened hind legs with seta and smooth, streamlined, and solid body. Dytiscidae boast distinctive chemical properties, such as defensive secretions containing steroids not known in any other animal. For this reason, diving beetles have been a source for pharmaceutical company wood. In different parts of East Africa, young girls and boys prompt bites from the beetles for pubertal benefits, and for boys, to help them learn to whistle. Dytiscidae have also attracted study for notable parts of their evolution, including a sexual arms race, and their body size evolution following a rare early burst model. Ecologically, Dytiscidae's main limiting factors are anthropogenic activity, fish, and parasitic mites. Surface color and a sufficiency of aquatic plants are other influences on diving beetles' habitats. Due to being most common in unpolluted water, they can be a good water quality indicator. They can potentially control mosquito populations by feeding on larvae as well. They are able flyers so that they can colonize different habitats. Some species live up to several years, and most are unevolving with 2 to 3 month breeding periods. Various species overwinter, estivate, or enter deepest. In culture, the diving beetle is prominent in a Cherokee creation story. Without wasting any more time, let's jump into the fascinating world of habitus. Like most other water beetles, adult Dytiscidae have an oval habitus, often tapering toward the head with the pronotum widest at the base. Generally, it is smooth, flattened, and solid. Many species are only smooth macroscopically, though. The dorsum often has microsculpturing, including a mesh of loops, shagreen, lining, dotting, hatching, or granulation in combinations varying by species. The head, thorax, and abdomen are all streamlined, that is, they are integrated into a single, overall cohesive oval, as opposed to the 3 visibly articulate sections of some Carabidae like Brachynes. The elytra are so prominent that they conceal the abdominal sclerites. Now let's delve into the intricacies of head and explore its various aspects. The head is prognathous both in the adult and larval stages and wider than tall in shape. To a degree, it is drawn into the pronotum, whose pointed anterior, lateral angles abut the head's edge. The clitoris and fronds of the head are fused. Diving beetles largely conform to one mouthpiece model, with the following characteristics: a transverse upper lip with a notch in the middle of the anterior edged mandibles, apex forked into towards, sickle-shaped lacinia with apex pointed and covered with dense, strong hairs along the inner echinorodale, with 2 segments as in most other edaphiga, maxillary palpi with 4 segments and a developed palpida, and labial palpi with 3 large submentum apically expanded into lateral projections transverse, concave mentum with wide, upward curved lateral projections meeting the prementum transverse prementum with the anterior margin moderately notched and covered with long seatanage on the mentum's anteromedial edge forming a median projection and a notch on the projection. As we move forward, let's uncover the untold stories and fascinating intricacies of abdomen. Diving beetles across subfamilies have a set of 6 abdominal segments, or ventrites, visible on the belly. They are lined up roughly perpendicularly to the sagittal plane, one after the other. The segments can occupy much of the abdomen's ventral side, from the anterior to the posterior. All the visible segments have different lengths in alignment with the oval shape's curvature. The last ventrite, on the anus, is the hypopygidium. It is modified to end in a notch, tubercle, or keel at the apex, and its surface texture is often wrinkly. Sometimes, the hypopygidium receives a process of the elytra's epipleura inserted into the former's pit. Accompanying the ventrites are 8 turgets and 8 variously sized pairs of spiracles for breathing. Also an identifying feature of the belly is the prosternal process. It usually projects back to the mesocoxa in a spear shape. The male atypicus is symmetrical, although Dytiscidae is among the beetle families whose atypicus experiences retornment longitudinal turning over development. The female ovipositor includes a pair of appendages connected with the hypopygidium called gonococcyx and basal sclerites called valvifers. The structure is set into the abdomen. With the groundwork laid, let's now examine legs and its connections to our previous discussions. The 4 anterior legs are rather short, and the front pair is slightly flattened, whereas the hind legs are enlarged and strongly flattened for swimming. Generally, the legs' front tibiae lack spines. The tarsi have a tarsal formula of 5-5-5 and either 1 or 2 claws. Species with 2 claws have their mesoscutellum large and exposed. The claws are not toothed and can either be equal or unequal in size. The fore and middle tarsi have 4 segments, with the 4th sometimes very small and concealed between the 3rd segment's lobes. As we progress, let's zoom in on identification and examine its role in shaping our overall narrative. Unlike Notoridae and most Hydrophilidae, the dorsal surface is not more strongly convex than the ventral. Dytiscidae also differ from Notoridae in their mandibles, since they lack enlarged portions there of the molars. The antennae are glabrous and number up to 11 depending on the species. Dytiscid antennae are thread-like, generally longer than the head's width, and look like their palps except longer, whereas hydrophilid and gyrinid antennae are clubbed. Their eyes are flat rather than protruding out, and, unlike Gyrinnidae, are not divided. Unlike Carabidae, they do not have a transverse suture on their metasternum. They also lack the keel-like feature between the legs of many hydrophilids. All known diving beetles except ones in the genus Selena have the scutellum concealed, with only a minuscule part seen from the surface. This does not mean that the scutellum is invisible as in Notoridae. Moving ahead, let's uncover the hidden gems within sexual dimorphism and discover their significance. Many species in Dytiscidae are sexually dimorphic. Males have suction cup-like palatus on their legs to help them keep grip of females during copulation, and, in many species, females have furrows on their elytra and variously the pronotum and base of the head. In males, these parts are smooth. The furrows of the female uneven the elytral structure, interfering with the male's grip. They weaken it likely with the aim of increasing the female's control over mating. Brace yourself for an in-depth analysis as we navigate through size and its far-reaching implications. The length of adults averages at 1 to 2.5 cm, and the elongate larvae range from 1 to 5 cm 0.39 to 1.97 in. The largest known adults are of the species Megadytis ducalis, at up to 4.75 cm 1.9 in, and Dytiscus latissimus, at up to 4.5 1.8 in. At about 0.9 0.035 in. Meanwhile, Limbodessus atypically is likely the smallest. Let's now shift gears and explore color through a critical lens, uncovering its strengths and weaknesses. Most are dark brown, blackish, or dark olive in color. Diving beetles in some subfamilies sport golden highlights on the dorsal borders, others variously colored spots or bands. Sometimes, diving beetles are reflected with a metallic appearance. As we transition to the next segment, let's unravel the mysteries surrounding roller body shape and gain a fresh perspective. Diving beetles' shape is optimized to ease navigation through water by reducing drag and improving stability while swimming. No segment moves or bends off balance due to being integrated with the others. This reduces form drag in two ways. 1. It minimizes frontal resistance, which can upturn a swimmer with an uneven position. 2. It minimizes eddies, or waves, from eddy resistance that excessive, especially jerky, movement can incur. As they swim, diving beetles further streamline their bodies by tucking their foreformer legs into well-fitting grooves. Their body's water resistance differs considerably by hunting type, such as search or ambush. Now, let's shift our perspective and explore oxygen from a different angle. Like other water beetles, adult Dytiscidae get their oxygen while swimming by storing air in a space between their elytra and abdomen. At the same time, they can also diffuse dissolved oxygen from the water. The former ability keeps Dytiscidae alive underwater for about 30 minutes, whereas the two combined can give them around hour's worth of oxygen in one go. Some Dytiscidae have an additional way, using their elytra as a respiratory organ. One of them, Dytiscidae, has been recorded to survive 6 weeks without atmospheric oxygen. Dytiscidae and other smaller hydroporina have specialized seta on the elytra, pronotum, and ventral side that act as tracheal gills. The seta form a layer that keeps out water and traps air. Smaller species like these can also stay underwater for weeks because they can live off oxygen from natural vegetation. Another feature acting as a gill in diving beetles is a small air bubble pressed out from the subaliteral cavity and held by the hydrofuge hairs at the tip of the abdomen. The bubble shrinks over time, requiring the beetles to surface periodically due to gas exchange decreasing. This behavior of alternating between the surface and high depths is why they are known as diving beetles. When bigger species break the surface, they hang on it with their rear end protruding slightly. Along with regaining their bubble, they can also bend their abdomen slightly downwards for gas exchange in the subaliteral opening. Since larvae lack ventral gills, larvae instead have a siphon at the tip of the abdomen that they draw in air with. As we transition to the next segment, let's unravel the mysteries surrounding roll of hind legs and gain a fresh perspective. Their two hind legs are much larger and wider than the other two pairs of legs so that they can use them as oars or paddles and move faster. The seta on the legs are there to help them change direction quickly while swimming. They can swim both forwards and in reverse effectively without needing to rotate. Unlike Hydrophilidae, their hind legs move in synchrony while swimming, namely during the forward and backward motions. The legs move asymmetrically while turning, in opposing directions, to steer the beetle sideways. Unlike other aquatic animals such as turtles, jellyfish, fish, and frogs, they both can stay in one direction while retreating and have a lower turning radius when they do turn. These strengths are a testament to their superior flexibility. In the next segment, we'll be exploring process and its implications for our subject matter. Diving beetles' swimming process as a whole cycles between two steps together equivalent to rowing, the power stroke and the recovery stroke. The power stroke's function is to increase propulsion by means of maximizing the beetle's cross-sectional area and involves two actions. 1. The tarsi stretch out, as a result also rotating the connected tibiae longitudinally and flattening them. This use of the segmented legs gives diving beetles an advantage over human rowers when it comes to straightening the legs. 2. The seta on the tibiae bristle out and maximize water resistance. Because of the thrust that the hind legs give, the speed of each cycle peaks in this stroke. This peak spans 60% of the stroke according to the findings of Key et al. 2021. In the recovery stroke, the beetle then reduces the water resistance with an equalizing effect by rotating its tarsi and folding the seta flat. The same study finds that each cycle lasts for about 272 milliseconds, and the power stroke takes up about 47% of it. Most of the acceleration happens in the first 50 milliseconds. Likely for the purpose of escaping from predators, the acceleration is especially high when swimming backwards, with the increase in speed recorded to be from 0 to 27.3 km. In 25 milliseconds, the average acceleration is 9.8 km, whereas the average of the whole first 50 milliseconds of each forward swimming cycle is about 1.68 km. The speed of the forward swimming cycle is on average about 8.74 km, maximum 12.9 km, and minimum 5.69 km. The angular velocity of the turning cycle is on average about 8.3 rads, maximum 12.9 rads, and minimum 0.83 rads. With our foundation established, it's time to explore use of rectum underwater and its relevance to our overarching theme. An ability specific to the smaller of the diving beetles is to rapidly blast ingested water out from the rectum. This is a solution to water surface tension, impeding them from leaving the water to fly up away from it. On top of that, the rectal ampulla serves as a hydrostatic organ to regulate underwater buoyancy. To decrease buoyancy, diving beetles ingest water. To increase it, they expel water from their rectum like they do against surface tension. Diving beetles strategically adjust their fill to the optimal buoyancy over changing conditions. The goal is for the body to have approximately the same specific gravity as the water. In the following segment, we'll be examining chemical senses in greater detail. The chemical senses of diving beetles, smell and taste, are strong. These senses provide for their need to identify potential food. Their taste receptors are concentrated on the maxillary and labial palpi, and they can detect sweet, sour, salty, and bitter chemicals. Then, the antennal surface is where the receptors for smell occupy. For males, this surface doubles as a way to locate female conspecifics ready to mate. Females have the ability to secrete olfactory pheromones attracting males within an area of. This form of sexual signaling has been speculatively connected with the expanded antennamus seen in the males of many groups in Dytiscidae. Diving beetles are attracted to alarm pheromones emitted by fish that they eat as prey. In this way, the pheromones work against the fish as chaeromones. With our curiosity piqued, let's embark on a dedicated exploration of chemical and its fascinating intricacies. From their pygidial gland, medium and large-sized species can secrete two types of substances, one a fluid and the other a paste-like solid. Oftentimes they go above water to groom themselves with their secretions, especially the paste, and distribute them on their body surfaces. They are an antimicrobial safeguard, protecting against bacteria and ciliates. Underwater, diving beetles apply them to sensitive body parts like spiraculi and subliteral togal respiratory surfaces to protect them from water. Chemically, the secretion grooming paste consists of benzoic acid, a glycoprotein, and some phenols, particularly methyl p-hydroxybenzoate and p-hydroxybenzaldehyde. Until the secretions are released, they stay in a reservoir within the pygidial gland. The reservoirs are covered with muscle layers so that the muscles can move them out when it is time. In conjunction with the pygidial glands are the prothrosic glands, another source of defensive secretions. The prothrosic glands' reservoirs are not covered with muscle layers unlike the pygidial glands. Instead, diving beetles use internal turgor pressure and contract their turgosternal muscles. Once the secretions leave the reservoirs, they are discharged by way of one muscle that has its origin on the cervical membrane. Besides managing surface tension and buoyancy, the rectal ampulla is also a source of defense. When disturbed, diving beetles have the option to release odorous food residues from there to deter any organisms. Chemical defenses combat not only against parasites, but also predators. Steroids in the secretions can force a predator such as fish to regurgitate the beetle. A kind especially prevalent in diving beetles is predanins, such as 11-deuterocorticosterone. Experiments have shown predanins to possibly deter fish, protecting diving beetles from predation. Diving beetles are known to be the only family in the whole animal kingdom to produce certain kinds of steroids. For this reason, they are an important source for pharmaceutical wood. Chemicals in diving beetle secretions can also anesthetize or even kill predators. Other chemicals produced include a variety of aromatic esters, acids, and aldehydes. In this section, we'll be peeling back the layers of other to reveal its true essence. Small species do not have chemical defenses, so instead opt to avoid danger by reducing their activity underwater or dispersing themselves when in groups. Diving beetles can also defend themselves by playing dead thanatuses. Species hide, escape, and bite as well. Larger species such as Cybistrina and Dytiscidae kick with their hind legs. In the upcoming section, we'll be dissecting habitat and exploring its intricate connections to our topic. Diving beetles are the most diverse beetles in the aquatic environment and can be found in almost every kind of freshwater habitat, from small rock pools to big lakes. Some Dytiscid species are also found in brackish water. Diving beetles live in water bodies in various landscapes, including agricultural and urban landscapes. Some species, such as Agabus uligiensis and Acilius canaliculatus, are found to be relatively tolerant to recent urbanization. One of the most important limiting factors for diving beetle occurrence is the presence of fish, which predate on the beetles mostly on larvae, compete for food, and change the structure of the habitat. The presence or absence of fish can also affect habitat use and habitat selection of Dytiscids. Some species, such as Aurodytis sammarki, occur in exposed areas of waters, whereas many diving beetles species prefer habitats with aquatic plants, especially plants with complex structures, such as sedges and bulrush. Like other insects, their presence can be a good indicator of water quality. Oftentimes, surface color is a determiner of a diving beetle's water environment. Having bright colors with markings is tied to clear waters with mineral substrates, and being melanin high or green to habitats with dark substrates or dense vegetation. Some diving beetles live in areas in seasonal droughts. In these cases, they respond by either entering a terrestrial diapus or burrow into the stream substratum, often down to. The burrowing solution is likely the reason for their high survival rates following droughts. In addition, they can likely estivate over summertime dry spells. When they need to colonize a new habitat for mating or better conditions, they fly and look for light reflections from the water surface. In urban areas, diving beetles' attraction to lights draws them erroneously to artificial lights and glossy surfaces on cars, and so on. Meanwhile, on land, the gait of many adult diving beetles can appear awkward or clumsy due to their enlarged hind legs. As we venture forward, let's examine specializations in detail and gain a deeper appreciation for its significance. A special stygobatic variety of Dytiscids only live underground. Habitats include pitch-dark wells, boreholes, and caves. Most stygobatic species are in the subfamily Hydroporina. However, Exosalina abdita and Copilatus cesana have been discovered as among the exceptions. Select Hydroporina species live in terrestrial habitats, such as dry forest floor depressions or leaf litter, at least in the adult stage. Stygobatic species are prevalent in Western Australia because of the groundwater coming from its large network of paleodrainages. There, the beetles have been recorded to live in groundwater estuaries of salt lakes and shallow calcratis. Some species in Africophilus, Agabus, Fontidicus, Hydroporus, Hydrotrupes, and Platinetis are specialized for living in hygropetic habitats. Some, such as Hydroporus sardomontanus, are semi-hygropetric. Another less common environment type is interstitial or semi-subterranean habitats, such as gravel banks along rivers. Examples of interstitial species include Exosalina salucholsensis, Agabus paludicus, and Hydroporus bithynicus. Some of the stylobitic, interstitial, and terrestrial dytiscids have depigmentation and reduced or, in stylobitic species, none at all. Terrestrial species tend to also be smaller and have no seta on the mid and hind legs due to not swimming. Stylobitic species have fused elytra and an absence of wings. Interstitial species can have long sensory seta and reduced wings. Prepare yourself for an eye-opening discussion on diet in the upcoming portion of this video. Similar to their wide range of habitats, Dytiscidae can be massive generalists diet-wise. Predaceous diving beetles' diet can include both invertebrates and vertebrates. The larvae, especially, take on animals with the same or bigger size, such as fish and tadpoles. Adults readily eat both living animals and carrion, making them scavengers and water cleaners. In addition, diving beetles practice cannibalism, both within their species and outside it. Since they can feed on mosquito larvae, they have a potential role in biological control of mosquito populations similar to Hydrophilidae. In the next portion, we'll be immersing ourselves in the realm of larvae and examining its broader implications. When still in larval form, the beetles vary in size from about 1 to. The larval bodies are shaped like crescents, with the tail long and covered with thin hairs. 6 legs protrude from along the thorax, which also sports the same thin hairs. The head is flat and square, with a pair of long, large, and pincer-like mandibles. It looks like a capsule due to its sclerotization. Larva's eyes are stemata rather than compound eyes like the adults. During the first insta, larvae have 2 egg bursters on either side of the frontotepues. They use these to break out of their egg. Along with the mandibles, the mouthpiece is also made up of a maxilla and a labium. The maxilla is further made up of a cardo, stipes, a palp of 3 palpemers, and a palpiform galea. The labium has a postmentum on the base, prementum on the apex, and, attached to the prementum via a small palpega, a pair of labial palps. When hunting, they cling to grasses or pieces of wood along the bottom, and hold perfectly still until prey passes by. Then they lunge, trapping their prey between their front legs and biting down with their pincers. The larvae are also known to partially consume prey and discard the carcass if another potential prey swims nearby. Their usual prey includes tadpoles and glassworms, among other smaller water-dwelling creatures. Larvae in many species do not eat prey through their mouth opening, unlike larvae in other species and adults, because it is closed. Instead, they liquefy their food by injecting digestive enzymes, namely protease, through a canal opening outward near their pincer's tip. Once the whole edible portion is in liquid form, larvae ingest it with a sucking pump traversing the pharynx and cybarium. Because they digest their food before ingesting it, they are saprobiants and digest their food extracellularly. Adults, meanwhile, bite and swallow whole small sections of their prey at a time. The dorsal surface is usually distinctly sclerotized, like the head, but not the ventral surface. Their sclerotized plates only appear sometimes on the most posterior segments, while the rest of the surface is mostly membranous. Sclerite's pigmentation makes them often stand out from the rest of the body. The thorax has 3 segments, the pro, meso, and metathorax, whereas the subcylindrical abdomen has 8 visible segments. Each of the thorax's segments have a pair of articulated legs, a large turgid and, in most specimens, a pair of smaller lateral turgids associated with each leg attachment. On the abdomen, the first 7 are relatively uniform in appearance, while segment 8 is modified for respiration in varying ways. This last segment ends in a pair of orogompi. Some larvae are heavier than water, while others are buoyant and have to push to stay submerged underwater. The heavier larvae mostly move through creeping and burrowing and reach the surface by climbing or swimming. Turning our focus to development, let's explore its key elements. As the larvae mature, they crawl from the water on the sturdy legs and bury themselves in the mud for pupation. After about a week, or longer in some species, they emerge from the mud as adults. Adult diving beetles have been found to oviposit their eggs within frog spawn in highly ephemeral habitats, with their eggs hatching within 24 hours after the frogs and the larvae voraciously predating on the recently hatched tadpoles. Before pupating, larvae molt usually 3 times, correlating to their number of instars. If a flood occurs during pupation, there is a risk of drowning. Others deposit eggs in moist soil or debris by the shore, plant surfaces, or slits that they make in aquatic plant stems beneath the water. In the latter case, larvae in the first instar are usually attached to a plant and independent of surface air. For them, the plant serves as their substrate. The ability of adults to cut into plants with their ovipositor is unique to the genera Agabus, Coptotomus, Cybista, Dytiscus, Hyaticus, Olybius, Lacophyllus, and Thermonectus. This method could be superior to depositing the eggs in soil because plants typically stay moist much longer than surface soil does. In other cases, larvae diffuse oxygen throughout their body surface when underwater. The second and third instars take in surface air through a siphon at the abdomen's tip. Let's now enter the realm of life cycle and discover the fascinating stories it has to tell. Only one generation of diving beetles tends to be born every year and evolve in. Species of diving beetles can live up to around 5 or several years in total. Mating season for most species spans 2 to 3 months, most often within the time of spring and autumn, since adult diving beetles in many species overwinter. In habitats where the waterbed stays unfrozen, they hibernate in plant material and sediments at the bottom. Different species can also overwinter as eggs and larvae. Brace yourself for a deep dive into edibility as we explore its impact and relevance in our evolving narrative. Adult Dytiscidae, particularly of the genus Cybista, are edible. Remnants of Celsius Exponitus were found in prehistoric human coprolites in a Nevada cave, likely sourced from the Humboldt Sink. In Mexico, Celsius Exponitus is eaten roasted and salted to accompany tacos. In Japan, Celsius Japonicus has been used as food in certain regions such as Nagano Prefecture. In the Guangdong province of China, the latter species, as well as Celsius bengalensis Celsius, Guarani Celsius, Limbodessus Celsius, Sugulatus Celsius, Tripunctitus, and probably also the well-known great diving beetle D. marginalis are bred for human consumption, though as they are cumbersome to raise due to their carnivorous habit and have a fairly bland though apparently not offensive taste and little meat, this is decreasing. Dytiscidae are reportedly also eaten in Taiwan, Thailand, and New Guinea. Get ready to immerse yourself in the world of as pets as we examine its impact and relevance. Diving beetles can be kept in a water tank as pets. The time has come to unravel the secrets behind diving beetle conservation and gain a deeper understanding. The greatest threat to diving beetles is the degradation and disappearance of their habitats due to anthropogenic activities. For example, urbanization has led to the decreasing quantity and quality of Dytiscid habitats, which consequentially has increased the distance between habitats. Thus, Dytiscids may be exposed to high predation risks during dispersal. The negative effects of urbanization on Dytiscid communities can be long-lasting from the temporal perspective. Some species may go extinct across an urban landscape in the long term, resulting in further losses of urban biodiversity. Urbanization has complex effects on the inter- and intraspecific variation in Dytiscid traits. Some flight-related traits of Acilius canaliculatus and Hydaticus seminiger, such as body length and hindwing traits, were found to change along the urban gradient at different scales, whereas the traits of Olybius eta exhibited no change. Brownification, which refers to the change in surface water color towards yellow-brown hues caused by recent climate change and land use change, can also drive changes in Dytiscid communities. As some species, such as Dytiscus marginalis, are tolerant to brown water, whereas some species, Hyphidrus ovatus, tend to occur in clear water. Brownification may threaten Dytiscid species that are intolerant to highly colored waters. Drainage can have adverse effects on their populations. For example, species such as Rantus bistreatus and Graphoderus bilinitus went extinct in Britain likely because of the drainage of the Whittlesea Mere. Drainage affects Dytiscidae mostly due to disturbing their breeding cycles, as demonstrated through how Dytiscid populations dramatically increased in East Asian paddy fields. Their flourishing started after rice producers switched from the conventional method of draining the land mid-season while it is flooded, to no-till. Dytiscid adults are eaten by many birds, mammals, reptiles, and other vertebrate predators, despite their arsenal of chemical defenses. But by far the most important predator of diving beetles are fish, which limit the occurrence of most diving beetle species to fishless ponds, or to margins of aquatic habitats. Although the larvae of a few Dytiscid species may become apex predators in small ponds, their presence is also often incompatible with fish. Therefore, the main focus of water beetle conservation is the protection of natural, fishless habitats. In the European Union, 2 species of diving beetles are protected by the Berne Convention on the Conservation of European Wildlife and Natural Habitats, and thus serve as umbrella species for the protection of natural aquatic habitats, Dytiscus latissimus and Graphoderus bilinitus. In the next portion, we'll be immersing ourselves in the realm of cultural significance and examining its broader implications. The diving beetle plays a role in a Cherokee creation story. According to the narrative, upon finding nowhere to rest in the liquid chaos, the beetle brought up soft mud from the bottom. This mud then spread out to form all of the land on Earth. Get ready for a captivating exploration as we unravel the layers of ethnobiology and their profound significance. Adult Dytiscidae, as well as Geirinidae, are collected by young girls in East Africa. It is believed that inducing the beetles to bite the nipples will stimulate breast growth. The effect of that habit has not been tested, but it is notable that the pygidial and prothoracic defense glands of diving beetles contain many types of bioactive steroids. The steroids that are operative include estrone, estradiol, and testosterone. In Uganda, girls do not use Dytiscidae, but only the smaller Geirinidae, since it is believed that the Geirinidae are the females and the Dytiscidae are the males of the same species. Beetles in these 2 families are known as Urinet mother of water, Amharic in Tanzania and rural regions of Ethiopia. For the opposite effect, young boys in Tanzania's Njom region use the same technique. They do so to mitigate the breast growth that can temporarily arise during the period of puberty before testosterone levels go up. Meanwhile, in other areas of East Africa such as Zimbabwe, diving beetles are an aid for boys learning to whistle. In this case, the beetle bites the tongue. Now, let's shift our perspective and explore parasites from a different angle. Dytiscidae are parasitic by various mites. Those in genera Dytiscurus and Iles live beneath the elytra of their hosts, those in genus Acheronticurus attach to the mesosternal regions, and those in genus Hydrotina attach to various locations. These mites are parasitic as larvae with the exception of Dytiscurus, which are parasitic for their entire life cycle. Now, let's delve into the intricacies of phylogeny and explore its various aspects. Here is a simplified cladogram based on the results of Kelvin B. Miller and J. Bergston's 2023 analysis of taxon gene samples using the parsimony and Bayesian models. This cladogram goes down to the level of the subfamily, and only includes the 11 that are extant. The Agabinus subfamily proved paraphyletic, therefore the relationship does not apply to some of its genera, namely Hydrotrubes and Platanectis. These 2 deviate away from the Agabina plus Colymbetina grouping. Here is a version of the cladogram including the merely tentative relationships whose support from the analysis was not strong enough to be conclusive. These are the grouping of Lancentina with Agabina plus Colymbetina and Coptotomina with Hydroporina plus Hydroditina. Prepare yourself for an in-depth analysis of evolution in this section. As observed in both sexually antagonistic morphology C-shape and morphology and behaviors, females in Dytiscidae are more selective when it comes to mating. One behavior is a technique to break the male's grip. When approached by males, females in some species make fast and erratic swimming movements. As in most insects, mating tends to be more deliberate for females because offspring have a much higher energy cost for them. With this cost comes reduced fitness to mate again. Under an evolutionary lens, an evolutionary sequence called the arms race explains that females first evolved resistive behaviors to minimize their losses. Next, males' morphology evolved palates to stop females from escaping. Then came the female's textured dorsal surface as a counter-adaptation. So, female morphological adaptations came before behaviors according to this model. It is called the arms race because it involves either sex continuously one-upping the other's previous adaptation. According to Kelvin B. Miller's 2002 cladistics analysis, the male's palatus originated in Dytiscidae. 5 groups within Dytiscidae then evolved the female dorsal surface, each independently. Everson et al. 2019 describe a standstill in species evolution as a byproduct of the Dytiscid arms race, specifically in Graphoderus zonatus, in contrast with sexual conflict normally being associated with divergence and diversification. There is a clear consensus that diving beetles' ancestors were terrestrial and similar to ground beetles. Testing found that the body size evolution followed a model very rare among animal groups. The model consists of an early burst period of rapid morphological change in many Dytiscids transitioning from lentic to lotic habitats, then a long static period of phylogenetic conservatism. However, this early burst did not seem to correlate with any species diversification. This indicates that morphology and species diversification are uncoupled in Dytiscidae. In the next portion, we'll be immersing ourselves in the realm of systematics and examining its broader implications. The following taxonomic sequence gives the subfamilies, their associated genera. Subfamily Agabina Thomson, Agabinus crotch, 1873 Agabus leech, 1817 Agametris sharp, 1882 and Nettis goekweave, 1971 Hydronebrius Yakovlev, 1897 Hydrotroops sharp, 1882 Elibisoma crotch, 1873 Elibius Erickson, 1832 Laurinectis sharp, 1882 Platimus Thomson, 1859 Platinectis regimbart, 1879. Subfamily Calimbetina Erickson, Anisomeria brink, 1943 Senilites brink, 1948 Karabastau Balk, Hendrick and Walker, 1992 Bunitz Spangler, 1972 Calimbetis Clairville, 1806 Hoprius fall, 1927 Meladima Laporte, 1835 Melanoditis Seidlitz, 1887 Neoscutoprus Balfour Brown, 1943 Rantus de Jean, 1833 Rugosus Garca, 2001. Subfamily Copulatina Brandon, Agopromarthus Zimmermann, 1921 Aglimbius Sharp, 1880 Copulatus Erickson, 1832 Exocelina Bruin, 1886 Lacinectus Mochalski, 1855 Leopterus de Jean, 1833 Madaglimbius Shaverdo and Balk, 2008 Rugosus Garca, 2001. Subfamily Coptotomina Brandon, Acoptotomus Say, 1830. Subfamily Cybistrina Straditis Watts, 1978 Bifurcitus Brink, 1945 Cybister Curtis, 1827 Nilsonitis Miller, Mickett, and Ferreira Jr., 2024 Megaditis Sharp, 1882 Methaxiditis Miller, Mickett, and Ferreira Jr., 2024 Paramegaditis Tremoules and Bockman, 1980 Anicahydrus Shome and White, 1847 Regimbartini Chattanay, 1911 Spenceratris Sharp, 1882 Stenhydrus Brink, 1945 Trifurcitus Brink, 1945. Subfamily Dytiscina Leech, Basilius Leech, 1817 Athionectus Sharp, 1882 Astroditis Watts, 1978 Dytiscus Linus, 1758 Aretus Laporte, 1833 Graphoderus de Jean, 1833 Hydesicus Leech, 1817 Hydroides Hope, 1838 Megaditis Sharp, 1882 Dytiscus Wickham, 1911 Nototicus Zimmermann, 1928 Anicahydrus Shome and White, 1847 Regimbartina Chattanay, 1911 Rantaticus Sharp, 1880 Sandricatus Sharp, 1882 Spencertrus Sharp, 1882 Stenhydrus Brink, 1945 Thermonectus Digene, 1833 Tpholutions Omer Cooper, 1956 Ambarticus Yang et al. 2019 Burmese Amber, Myanmar, Late Cretaceous Cenomanian. Subfamily Hydrodictyna Miller, Hydroditis Miller, 2001 Microhydroditis Miller, 2002. Subfamily Hydroprena orb, Ifracoditis bistrum, 1988 Agnishydrus bistrum, Nilsson and Walter, 1997 Alidissus quidnit, 1953 Elipocrea Zimmermann, 1924 Amoriditis Regimbart, 1900 Amoriditis Ferry and Petrov, 2013 Andix Sharp, 1882 Anginipocrea Walter, Belk and Hendrick, 2001 Another Tylis Babington, 1841 Antiporus Sharp, 1882 Barrett-Thydrus Lee, 1927 Bytisodis Regimbart, 1895 Bytisonitus Regimbart, 1895 Bytissus Sharp, 1882 Boronetis Angus, 2010 Bornodissus Belk, Hendrick, Mazzoldi and Bistrom, 2002 Brachyvatus Zimmermann, 1919 Branchoporus Hendrick, Toussaint and Belk, 2014 Calthiporus Zimmermann, 1919 Carbidrus Watts, 1978 Selena Orb, 1837 Chostanectis Sharp, 1880 Clypoditis Regimbart, 1894 Colhydrus Sharp, 1882 Camaldussus Spangler and Barr, 1995 Crenodussus KB Miller, 1997 Darwin Hydrus Sharp, 1882 Dronectus Sharp, 1882 Derivatilis Sharp, 1882 Desmopocrea Babington, 1841 Dimishydrus Uno, 1996 Erboporus KB Miller, Gibson and Alary, 2009 Etruscoditis Mazza, Ciabfoli and Roki, 2013 Fomtidissus KB Miller and Spangler, 2008 Judicus Brancucci, 1979 Gibidissus Watts, 1978 Glaridissus Walker and Bistrom, 1998 Graptoditis Sidits, 1887 Heidiporus Young and Longley, 1976 Hemabidissus Zimmermann, 1921 Herisos Gwidnit, 1949 Herphidrus Sharp, 1880 Heterodrus Vermeer, 1869 Heterostenuta Strand, 1935 Hoverhydrus Bistrom, 1982 Huxelhydrus Sharp, 1882 Hydroculus Rowley and Larsen in Larsen, Alary and Rowley, 2000 Hydrodissus J. Belfor Brown, 1953 Hydroglyphus Mochalski, 1853 Hydropepus Sharp, 1882 Hydroporus Tvervel, 1806 Hydrovitus Mochalski, 1853 Hygretus Stevens, 1828 Hyphoporus Sharp, 1880 Hyphobatus Water and Bistrom, 1994 Hyphidrus Elijah, 1802 Hypodissus Gwidnit, 1939 Iberoporus Castro and Delgado, 2001 Incomptodissus KB Miller and Varka, 2011 Cacodudissus Hendrick and Belk, 2009 Cachalidrus Ordish, 1976 Lacanellus Rowley and Wolf, 1987 Lacanis Gozes, 1914 Lioditis Gwidnit, 1936 Limbodessus Gwidnit, 1939 Lidissus Gwidnit, 1939 Dioprius Gwidnit, 1950 Megaporus Brink, 1943 Metaporus Gwidnit, 1945 Methylis Sharp, 1882 Microdissus Young, 1967 Microditis J. Balfour Brown, 1946 Morimoto Uno, 1957 Nebrioporus Regimbart, 1906 Necrosoma W. J. McPhee, 1871 Nobidisodis Hendrick and Belk, 2009 Nobidissus Young, 1967 New Flypoditis Young, 1967 Neuporus Gwidnit, 1931 Oroditis Sidits, 1887 Pachydrus Sharp, 1882 Pachinitis Regimbart, 1903 Papoidissus Belk, 2001 Porosta Sharp, 1882 Pechetius Gwidnit, 1942 Petrodissus KB Miller, 2012 Fretidissus Ordish, 1976 Platoditis Bistrom, 1988 Pedrus Gwidnit, 1945 Primus Sharp, 1882 Pseudovirus bistrum, 1988 Sacropomporus gene, Tales and K.B. Miller, 2012 Pteroporus guidnit, 1933 Quedus sharp, 1882 Rithriditis Bamiel, 1989 Sanphilippiditis franciscoe, 1979 Scarditis doses, 1914 Schistomorus palma, 1957 Cephaloporus watts, 1997 Schaphidrus elmer cooper, 1958 Cyanoporus spangler, 1996 Dytiscidae bile de Perrin, 1904 Cyanodytis spangler, 1996 Spangler Lorodosus K.B. Miller and Varka, 2011 Stenopriscus sharp, 1880 Stytonectus brink, 1943 Stytotussus Zimmermann, 1919 Styloporus Larsen and Lebomte, 1994 Suffroditis doses, 1914 Tepoidisus spangler, 1981 Terrestrial watts, 1982 Tiporus watts, 1985 Trichonectis guidnit, 1941 Troglodidnatus Sanfilippo, 1958 Tyndallhydrus sharp, 1882 Typhlodissus brancucci, 1985 Avarus guidnit, 1939 Vatulus old, 1837 Yoedoses, 1886 Yoena guidnit, 1936 Calicobacillus K.B. Miller and Lubkin, 2001 Procoambus the Bold, 1937. Subfamily Lacophyllinagistal, Ifricophilus guidnit, 1948 Agabatus crotch, 1873 Astralphilus watts, 1978 Japanoicophilus sat, 1972 Lacodytis regimbart, 1895 Lacophilus leech, 1815 Lacoporus j. Balfour Brown, 1939 Lacostinus brancucci, 1983 Napoditis steiner, 1981 Netostinus sharp, 1882 Phyllocolilus guidnit, 1937 Phyllocolilus guidnit, 1937 Phyllodytis j. Balfour Brown, 1939. Subfamily Lancetina Brandon, a Lancetis sharp, 1882. Subfamily Matina Brandon, Olomatus mauchamps, 1964 Batrachomatus Clark, 1863 Matisorb, 1836. Subfamily Ilidetes Acinaprokin and Wren, Lydraporus prokin and Wren, 2010 IUCN Formation, China, Early Cretaceous Action Lytiscus prokin and Wren, 2010 IUCN Formation, China, Action Mesoterus prokin and Wren, 2010 IUCN Formation, China, Action Lydixianus prokin, Petrov, B. Wang and Ponomarenko, 2013 IUCN Formation, China, Action Mesoditis prokin, Petrov, Wang and Ponomarenko, 2013 IUCN Formation, China, Action. Subfamily Dytiscidae Cretiditis Ponomarenko, 1977 Doranino Formation, Russia, Early Cretaceous Permian, Silza, Kazakhstan, Late Cretaceous Terrestrial Palaeodytes Ponomarenko, 1987 Karabastau Formation, Kazakhstan, Late Jurassic Oxfordian, Zozo Formation, Russia, Action Palaeodytes Incompletus Ponomarenko, Koram and Jarzembowski, 2005 Darlston Formation, United Kingdom, Early Cretaceous Variation, Undescribed Genus Cynoporus prokin and Wren, 2010 IUCN Formation, China, Action. Your feedback helps me improve, so please take a moment to leave a comment or review.